So welcome to the next session. The next session we're going to look at high throughput screening and its use in drug discovery. So the contents of this session is to look at what is meant by high throughput screening. We're going to look at the historical perspective of high throughput screening, of when it was discovered and when it was implemented. We're going to look at what is needed for high throughput screening, so things like looking at the biology, the chemistry, and where we need the biology and the chemistry to work together. Also, we're going to look at the hardware, so these are things like the dishes that we use in high throughput screening. And finally, we're going to look at what does the future of high throughput screening hold. So what is high throughput screening? Well, high throughput screening essentially is having kind of a large capacity for efficient repetition, and it involves a loss of repetition. It involves also the screening, so this is a process where we can perform an experiment to determine the nature of a material of use. And so therefore, high throughput screening is essentially a repetitive experimenting, and the experiments will be done thousands of times to find kind of a series of compounds, and this might be from like millions in the kind of in the bank that have a kind of a drug-like activity. So looking back at the kind of history of high throughput screening, it's only about 100 years old, and this is actually when the birth of kind of systematic drug research kind of came about. The first screen was done back in 1907 when it was discovered salvarsan for syphilis. And the well plate, which is commonly used today, actually originated from this guy, Dr. Tukatsky, in 1951. And Dr. Tukatsky was actually a leading scientist during a Hungarian flu epidemic. And what happened was that they wanted to kind of reduce the amount of equipment that they were using. So they wanted to reduce the microbiology equipment that they were using in the screening. And they came up with this well plate idea. 1 year later, Tukatsky then invented a 96 well plate, and this is actually something that we commonly use now in the lab. And the 96 well plate essentially is made up, it's in plastic, and it consists of 8 rows, normally labelled A down to H, and then there's also 12 columns across the well plate. So this is a general format of the 96 well plate. We're going to revisit some of the history around high throughput screening. So in 1961, Eppendorf released the first industrial manufactured piston stroke pipette, and it looked a bit like this. Okay, so it was had a fixed volume, so you couldn't change the volume like we can with the modern pipettes, but this was like the first pipette that was made by Eppendorf. And just to give you an idea, at the time, in 1961, the first silicon chip was manufactured by Kilby and Noyce, and it wasn't until 1971 that Hamilton produced the first digital diluter. Okay, and then in 1974, Gilson, who made this was the first model of Gilson. They patented a mechanical basis of an adjustable pipette, so we could actually change the volume. Okay, and it's using the piston idea, but we could change the volume, and that's what we use now in the lab, and Gilsons are kind of quite common now. In 1982, Infotech actually starts to build the first automated liquid handling workstation, and this allowed them to kind of work with plates and to dispense kind of automated liquid into each of the wells. And it wasn't until 1990 that Tomtech developed the first 96 channel liquid transfer device, which looked a bit like this. So you'd put your 96 well plate into these slots, and then it would dispense liquid into each of those wells. kind of looking at the multi-well plate. So high throughput screening was first introduced by pharma industry in the 1990s. And the first plate that was used was the 384 well plate, and that was made by Dr. G. Lennon in 1992. The 384 plate looks a bit like this, so you can see that there are rows from A to P, and then along the top we've got at least 24 columns going down. And SBS, which is known as the Society for Biomolecular Sciences, came up with a standard that all the plates would be standardized. Okay, so that was agreed in 1995, and what they did was they actually agreed on the distance between the wells, the height, and just the general kind of layout of the plate, and it would be kind of standardized between manufacturers. Sbs Standards, they're used globally by all major manufacturers, and they're used as like kind of robotics, multi-channel pipetters, pipette tips, reservoirs, plates. Mans and Young, also known as Wattman, developed the first 1,536 well plates. So you can notice they're getting bigger, okay, and that was back in 1996. And the same team then developed the next plate size up, which was 3,456 well plate. Oldenburg, also known as DuPont, they developed the 9,600 well plate in 1997, just a year later. And in the kind of early 21st century, acoustic transfer, so this is using kind of sound waves to transfer nanoliter across to the plates. And in 2016, at the ELRIC, it was an ELRIC conference, the Europeans, known as the European Laboratory Research and Innovative Group. This was a conference, and they actually showed that the 384 well plate is still the most popular format. So even though we have all these different formats, the 384 well plate is the most popular format. Just to give you an idea of the sizes of the volume that you can put into these plates. So and the effect of kind of greater density of the wells. So the 96 well plate has a typical assay volume of between 100 to 200 microliters. So normally 100 microliters is kind of the minimum volume. And this plate format can be used both by humans and also by robots. The 384, this has a volume, so we can reduce the volume. So this obviously saves reagents. So this is anything from 25 to 50 microliters. This can be used by humans and robots. The next size up is also a low volume 384. This is using much smaller volume, down to 5 microliters up to 20 microliters. Again, this plates can be used by humans and robots. Only just though. Whilst the next format, so we've got the 1,536 well plate. Now you can see that the volume is much smaller, so around about 2 to 10 microliters. So these are very small volumes, and it's actually very difficult to work with these types of plates, and so only robots can really use these. And that's the same is true for the 3,456. That uses 1 microliter, so definitely robots as well. And also the 9,600, which is very small volume, impossible to work with these volumes accurately. So that's the 0.2 microliters, and definitely robots for this one. And so you can see that when we reduce the volume size, this means that we use less reagents, and also it's then going to be cheaper. So it's going to be cheaper to do our experiments, so cheaper data points. And Affinac's actually rumoured to be making an even bigger plate, so 20,000 well plates, and that's using a volume of 25 nanoliters. So that's really small volume, so probably impossible to work with. So it's kind of a summary of the miniaturization. So you can see how we've kind of been miniaturizing these well plates. So we've gone from the 96 well plate, where we can screen around 10,000 compounds per day. And with a volume of 100 to 200 microliters, and this is around about the the mid 1990s. The 384 well plate, we can screen around about 40,000 compounds per day, and this is with a volume of 30 to 100 microliters. And the 1536 well plate, we can screen about 200,000 compounds per day, and this is a volume of 2.5 to 10 microliters. So there are some challenges of miniaturization, and these are kind of really down to kind of the physics. Okay, so we think about the pipetting volumes, so the physical properties of those liquids have a greater effect on kind of smaller volumes. Also evaporation, so we can get evaporation and sample loss. So we're working with really small volumes, then the sample loss will have a direct impact on the experiments. Also, you get surface tension, so the entire assay volumes can get stuck to the sides of the tips, so that can be an issue. And this shows kind of like a 5 microliter drop, which is hanging on the side of the tip, so that could be an issue, especially if we're using automated liquid handling. And this is due to kind of electrostatic forces that can move powders and liquids. So now we're going to look at what is needed for high throughput screening. So there are a number of steps involved in kind of high throughput screening. The first step is really we need to kind of discover what's our target on our phenotype. So we need to be able to use this to develop an assay. Okay, and we're going to focus on each of these steps. So we need to develop the assay. So assay development, so the drug discovery assays must be as simple as possible. So we want to use very few reagents in our assay. There also needs to be a minimal incubation time and also minimal mixing and also separation steps. It also needs to be cost effective, so it needs to be, because we want to screen lots of compounds, but we don't want it to be too expensive. Also, we need to have a large window, so this is the difference between positive and negative results, and this will come clearer in a moment. And the official term is known as wide dynamic range. We also need to have clearly interpretable results. So we need to be able to see the results and see whether or not there is an effect or there isn't effect. It needs to be easily automatable. So, and most things are actually, but this costs money, obviously. It needs to be robust, so it needs to be repeatable, not just by the robot, but also by different people. So confirming assay robustness, how do we do this? So high throughput scale experiments, this is kind of screening which requires repeatability, okay? And robustness is kind of an alternative term to assay the quality, so the quality of our experiment, okay? Or the quality of our data. And a high quality assay gives the same results every time, and that's without an error. And we can use something called the Z prime factor. So the Z prime factor is the industry standard for measuring robustness, and it's composed of this equation. So Z prime factor equals 1 minus this equation, which is 3 times the standard deviation of the positive control, plus 3 times the standard deviation of our negative control, divided by the mean of our positive control, take away the mean of our negative control. So if we do this calculation, so we need to have a really good positive and negative control, and they need to be kind of reproducible. So a perfect Z prime would be a Z prime of 1. This would be the perfect, and it would be excellent. whilst an acceptable Z prime would be anything above 0.5, whilst a Z prime that fails would be less than 0.5. So when we lay out the plate, so we were looking at the positive and negative controls, we lay out the plates, so essentially we have 4 sectors in the plate. So here we've got our positive control, so this is where we'll add our positive control, and in the other sectors we'll add our negative control. And we'll also make 3 replicates of this same plate, okay? So we don't just rely on one plate, we need to make multiple replicates, so 3 replicates, at least 3 replicates. We can actually monitor Z prime, and this can be done once you have an established assay robustness, and that needs to be kind of monitored, constantly monitored, and this can be incorporated into the screening. So the last 2 lanes, and the first 2 lanes, we can add our positive control, and here we can add our negative control. And in fact, most large farmers use these outside columns to monitor the Z prime in every plate. For example, Venom Tech actually have a thing called a targeted venom array, known as a TVDA, and they keep these columns empty, so for the customers to use, and then they can add their own negative and positive controls. So now we're going to look at the chemistry. So what is needed for high throughput screening? So if we look at the chemistry, so we've considered the development of the assay. We need to consider things like the plate type that we're going to use. Are we going to use the 96 well plate? Are we going to use the 384? We need to consider the number of cells that we're going to add to the wells, or if we're using protein as our screen, then we need to know how much protein we're going to add. Also the assay component, so what other things do we need to add, and how long do we need to incubate. If we're thinking about miniaturizing, so we can go from the 96 well plate to the 384. This may decrease the variation, and we can enhance the separation of signal. And we need to optimize that Z prime, okay? But if we think about the chemical library, so these are the synthetic and natural products. Now these feed into our high throughput screening. This is why we're doing the high throughput screening, is we need to have the chemical libraries. So what are the chemical libraries? Essentially the chemical libraries in many big pharmaceutical companies, they have stocks of libraries, and these are compounds which are kind of laid out in these racks in specially kind of controlled rooms, and here you'll have this robot which will move along and pick the chemical. So these are large collections of drug-like compounds which are kind of ready for screening, and these are called a library. Now, as I said, they're kept in this really kind of these large temperature-controlled warehouses. The compounds are all at the same concentration, and they're always being constantly added to. So essentially, they're going to build up big stocks of these compounds. And the aliquots have special barcodes on the end of the tubes, and they have a single barcode, okay? And the compounds will be dissolved in DMSO. And DMSO is ideal because it's a polar aprotic solvent, and it dissolves polar and non-polar agents. And this is particularly useful for stability of compounds. Now they use these specialized robots which are kind of working at a temperature to pick the tubes and assemble collections for dispensing into plates. This is all automated. If we think about some of the products, so we will look at like natural products. So these are like the first sources of the drugs. So these, and they still actually dominate most of the physician's arsenal. They come from things like microbes. So microbes, all of our actually current antibiotics have come from microbes, or slight modifications of the microbial compounds. So from penicillin onwards. They also come from complex mixtures that have been cleaned up, so deconvoluted, and then the synthesis needs then to be determined. Plants also contain or produce many metabolites. So we know about things like essential oils. Those essential oils can be kind of separated into individual compounds. Now, the complex mixtures from plants need to be separated, but in fact, plants have been shown to deliver many different drugs that we know about. So things like aspirin, Taxol, morphine, and others. And we also got peptides. So these peptides are essentially synthesized. Okay, so they have simple libraries, but usually they're only used for difficult assays, such as deorphination of G protein coupled receptors. So full versus target screen. So many large pharmaceutical companies have around 1 million compounds, and high-throughput facilities can actually screen an entire library in less than a month. In fact, now they can screen these compounds in 2 or 3 days. However, data analysis. So you need to do data analysis, you need to look at hit validation, and then you also have to follow up the hits. Okay, and this it takes a greater time, which then costs the high throughput screening. So if you have fewer, there's a higher value hits are actually preferred. There's a large cost of high throughput screening, which is the reagents and the consumables, and some robotic pipette tips are also quite expensive, anything between 50p to £1 each. So if you're thinking about scaling up, and if you're obviously testing million compounds, so you can imagine how much it's going to cost us to run the high throughput screening just in tips alone. What is trending now is the use of in silico methods. So this is using structural biology or structural modeling, and this helps us to narrow down compound libraries to produce targeted subset of a few 1,000 interesting compounds, which is very cost effective. And this is something that we're doing at the moment. We're using artificial intelligence in our screening process. So this is an in silico method to screen for potential compounds that will bind to our target. But this involves some information. So we need information about the structure of our target. So if we're looking at a protein, then we need the crystal structure. So to be able to develop or model our compounds against our target. So what sort of hardware is involved? So what is needed for the high throughput screening? So we need automation. So this automation of the assay, and this is really key in the process of high throughput screening, but also being able to screen many compounds quickly and effectively. So if we look at liquid handling, so liquid handling involves taking these compounds which we find in these vials and putting them into the plate. Okay, so we need to put them in a special order, okay, and this needs to be programmed into the robot to be able to add all these compounds into the plate in a specific way. So there are a number of different liquid handling tools for assay development. So the most simplest is the standard single channel pipette, which is still valuable in assay development. Also the manual multi-pipette, so this improves productivity. And we also have the automated multi-channel, which has a greater ergonomics. Other types of liquid handling tools are these small bench scale. So we've got the bench automation or replicators, also known as multi-drops, and also washers. So they're able to dispense small volumes into the plates. So why automate high throughput screening? If we consider cell culture, it takes many man hours to grow cells ready for screening. And by using liquid handling, if each well of a 384 well plate takes 10 seconds to pipette each reagent, a 2-reagent experiment would take 2 hours. And if you had 500,000 compounds to screen with 2 reagents, this would take 59 days, and that's working 24 hours, 7 days a week pipetting. So you imagine that would be really hard work. And if we're measuring, say, 1,000 in a 1,560 assay plate, this would take around about 52 hours of constant plate reading use. So taking the automated cell culture, this is kind of cell-based assays that can use between 200 and 200,000 cells per well. Here we can use hundreds of millions of cells, which are needed then for the high throughput screening. Here we've got an example. These are examples of, this is the CellMate system from the automation partnership. So this is an automated system for doing the cell culture. Here we have the Scalic T system from the automation partnership. Now both these systems are used for producing cells and also proteins for high throughput screening. And the Select can actually manage, so this one here can manage up to 168 cell lines in parallel. And that was actually designed for many drug companies, including Pfizer, GSK, Merck, Bristol Myers Squibb, and they produced batches of up to 300 assay-ready plates. If we take a look at the protocol to run the automated cell culture, on the Friday we would then get the flasks, the media, the cell stock, and the plates added to the carriers. And then after, the plates then would be incubated at 37 degrees, so this is the growing temperature of cells, especially of human cells, in 5% CO2. And as a kind of standard, the robot would then remove the flask from the incubator. So the incubator would be in the back here. And this is, you can probably just about see here, this is like the arm. The arm kind of reaches into the incubator and then removes the flask from the incubator. What it does then is it decants the media. So when we grow the cells, we always add special media, which helps to keep cells alive and gives them all the nutrients and food. So it decants it, and then it needs to then wash the cells. So the cells that we use will be kind of stuck down on the side of the flask, so it's on the bottom of the flask, so they attach, and then we need to kind of wash the cells. We don't want to wash them off, but we just want to wash away any of those dead cells, and then after we add then the trypsin. Now the trypsin is a protease, and this helps to detach the cells from the flask, so from the bottom of the flask. So afterwards we need to stop the trypsin. Okay, so we need to do this by adding full media, and that's with the FCS. So that's with something called fetal car serum, and that helps to stop or inactivate the trypsin, because we don't want that to work anymore. And then what the robot does then is it dispenses cells into a 384 well plate, and then it returns the plate to the incubator. To allow the cells then to attach, and they'll grow up over the weekend, and then on Monday morning the scientists then can remove the plates from plates then for high throughput screening. So this is an automated cell culture. This would be like a dream to have in the lab. We've also got the robotic liquid handling. So essentially these can be like special
is the latest
chambers
to the Hamilton
or
Robotics
cabinets,
portfolio. It is designed to easily automate and integrate your laboratory process all in one platform. The Vantage features cutting-edge pipetting channels, efficient and independent robotics, and plenty of space for labware, consumables, and third-party devices.
And then circulate.
Labware and samples are detected and identified during loading using the latest camera technology, thereby allowing full process control and tracking. In addition to labware detection and sample identification, the Vantage large volume range pipetting offers assay flexibility, while automatic transportation of labware minimizes programming efforts and increases assay efficiency. The Hamilton Vantage is designed to efficiently automate processes of all complexities. Besides automatic labware transportation, the Vantage also offers large storage capacity for tips and plates within the closed housing. The Vantage features multi-probe core heads in both 96 and 384 well formats. The compressed O-ring expansion technology secures disposable tips with minimal force, while the forceless tip eject ensures aerosol-free tip reuse or disposal in one of the definable waste compartments. Pressure and capacitive liquid level detection and pressure monitoring during each pipetting step ensures that the highest pipetting accuracy and reliability is met while maximizing sample safety. The Hamilton Vantage can accommodate up to 16 individual pipetting channels on one arm. Channels accommodate all sizes of Hamilton disposable tips. With increased positioning accuracy of the channels and arms, the labware that can be used on the Vantage has virtually no limit. Whether your application uses tubes, troughs, deep well, 96 well, or up to 1536 well plates, the Hamilton Vantage can address all labware and volumes with just one arm and one channel type. High hit-picking rates over plates or tubes is also achieved through the independent Y-spread of the channels. The completely new Nano Pulse channel offers a dynamic range of less than half a microliter up to 1,000 microliters. During a low-volume dispense, a microvalve built into the channel opens for a fraction of a second and releases just enough pressure to the liquid column underneath, releasing the exact amount of liquid defined. The entire process is monitored by a pressure sensor in the liquid channel. Aside from the Vantage hardware, which offers users physical safety, the Instinct 5 software further ensures process security. The software is accessible to varying user knowledge levels and has the flexibility to allow multiple users to run assays on one instrument or across a network of instruments. Instinct 5 brings an easier-than-ever graphical user interface to lab automation users. Through a clear visual interface, users can draw a laboratory process in a lab technician's language, while most values required to run the instrument are automatically programmed behind the scenes. Built-in dynamic scheduling maximizes instrument efficiency by running multiple activities in parallel. Users are also able to monitor the status of each assay in real time. Instinct 5 also helps minimize experiment setup time through 3D simulation of all robotic movements before and during a run. For custom programming needs and fine-tuned liquid handling, the Vantage offers users an advanced editor. Among the best in the industry, Instinct 5 provides multiple, traceable error recovery strategies and incident handling options designed to keep your assays running. The Hamilton Vantage also enables large integrations by offering expansion modules, including the track gripper and logistics cabinet. The expandable system design grants instrument access below deck for third-party devices, as well as provides tip and plate shuttles for increased sample throughput. To maximize lab space efficiency as well as to optimize pipetting and transportation activities, the Vantage features the track gripper. This vertical gripper moves independently of the pipetting arm and can reach any spot on or below the pipetting workspace in the logistics cabinet within seconds. Multiple levels of third-party devices can be integrated below deck, such as readers, washers, incubators, and shakers. The track gripper simply delivers labware below deck and the CELA-compliant device is automatically started. Once completed, the plate is moved to the next destination. In addition to third-party devices, the logistics cabinet can accommodate entry-exits which serve as shuttles for plates, tips, or even labware prepared for disposal. Magazines containing labware can be placed in any of 4 positions, holding up to 144 plates in one entry-exit. A barcode option can also be added for full sample and consumable traceability. In all, the Hamilton Vantage offers users integrated simplicity. From desktop solutions to large integrations, the Vantage optimizes lab space and instrument usage of entire laboratories. For more information, contact your local Hamilton representative today.
Now we have something called an acoustic liquid transfer system. This allows us to move liquids using something called special technology called acoustic liquid transfer. It's moving liquids with sound, which is really cool actually. So it can move very small volumes, so anything from 2.5 nanoliters up to 25 nanoliters, and that can be dispensed into these wells. And using sound waves, we can actually dispense liquids. That's pretty cool. So for the measurement, so this is the analysis, we use something called a plate reader. So a plate reader looks a bit like this. This is an example of a plate reader. Essentially, it can measure absorbance, but also can measure fluorescence. And all the assays need to have a measurable endpoint. So often we will measure fluorescence. So there are multiple types of readers for each type of assay. Okay, and we can use those different microplate formats, so the 96 well plate, the 384, the 1536 well plate. The serial connectivity, so this allows then for seamless integration, and you have accurate reproducible measurements, and also the same hardware which is usable in the assay. And there's also development to allow smooth transition to high throughput screening. So we can actually integrate the liquid handling and also the plate reader. Okay, so in this sort of like workbench format. So here we've got the liquid handling here, so the robot can dispense into the plates, and then obviously there's like an incubation going on, or it needs like we can actually even attach modules here, so we can attach an incubator. Afterwards, then we can read in the plate reader. So we can put all this together, okay, and we can take the robotic arms and move the plates between the module equipment, okay, so these are the robots which are kind of moving between bits of equipment, so between the incubator, the readers, so all you need is just one arm, but you'll need one arm to do most things, okay, so, but they can do that. So they can move between the incubators, the readers, the liquid handling stations, and this allows then for full hands-free automation. So what's the future of high throughput screening? Well, there's been a slowdown now in the drive to miniaturize, you'd be glad to know, but it's been 20 years now since the invention of the 1536 well plate, and as we said, the 384 well plate is actually still the most popular format. I think because it's the fact that humans and robots can work with this size well format. There's been a focus now on kind of high content screening, so kind of getting richer data, okay, so kind of using in silico screening, and this actually helps to save money, and then they only then wet screen the targeted subset library, so there's a big drive now to use in silico screening. Also, novel detection technologies to identify novel target classes. So we have a number of kind of established target classes already. So things like kinases, phosphatases, oxidoreductases, transferases, proteases, nuclear hormone receptors, G-protein coupled receptors, ion channels, and signaling pathways. Whilst there are a number of kind of novel targeting classes where we still need to kind of develop many compounds against. So we've got ion channels, transporters, transmembrane receptors, signaling pathways, protein-protein interactions, protein-RNA interactions, protein-DNA interactions, and DNA and RNA. And finally, some further reading to take a look at. So these are some references. The first reference is this book from Hill and Rang, Drug Discovery and Development, second edition. This is available as an ebook in the library, so please have a look at this. There's a couple of chapters there looking at high throughput screening. This is a resource looking at the history of the microplates. If you're interested to read up about the history of the microplates, have a look at this link. Also, plastics. So there's Plastics Historical Society, so the development of plastics for use in tissue culture. This is kind of like, you do use a lot of plastics. Have a look at this. SBS recommended microplate specifications. This is just a resource to take a look at. SLS Lab Autopedia, so looking at the history of automated liquid handling. Here there are kind of two articles. This is a review of cell culture automation. Have a look at this, and also academic screening goes through high throughput. This is quite a nice review. Anyway, thank you for listening. I look forward to seeing you in the session.